Welcome back. We are in the last of the lectures over muscle. This is the third lecture of Chapter 9. So far we've covered muscle structure. We've covered the details of muscle contraction with excitation-contraction coupling and the cross-bridge cycle. Today we're going to cover things that affect how strong a contraction is. So things that affect muscle contraction strength. When we're talking about strength of contraction, we're going to have to learn a couple of definitions, and we will look at a couple of different types of contractions. First, when we're talking about a muscle contraction, one word that we're going to hear a lot is tension. Tension is the actual contractile force generated by a muscle. And the other thing that we'll hear a lot about is load. And load, by definition, load is when a muscle is contracting and moving something. In our little diagram over here, it's a weight. Load would be how much force is applied to the muscle by the thing that's being moved. But for ease of explanation, we're going to actually just refer to load as what is being moved by the muscle. So for example, when you pick up your textbook or your laptop or anything that you pick up, your muscles in your upper arm contract, and they contract with a certain force that's called tension. And then the book or the laptop or whatever you pick up, in this case, would be the load. So the load is what you are trying to move. The tension is the force within your muscle. Over here we see two types of contractions. The first one here is an isotonic contraction. And what we see up at the top is kind of an experimental setup. We have a muscle, one end of it is anchored in place, and the other end of it is tied to a 3 kilogram mass. So here, the 3 kilogram mass represents the load. And experimentally, we apply some electricity to the muscle, and the muscle, in this case, contracts. It shortens, and the load is moved. In an isotonic contraction, tension within the muscle builds, the muscle shortens, moving the load. We can see here when the muscle is relaxed and the load is sitting on the ground, and then after we apply some sort of electrical stimulus, the muscle is much shorter and the load has been lifted. The graphs beneath show us tension within the muscle and length of the muscle. Here we see tension builds as the muscle contracts. And as the tension builds, the length shortens, and that's what moves the load. The other type is an isometric contraction. In this case, we see an experimental setup, the muscle still anchored in place, and the other end is attached to a 6 kilogram load. In this case, 6 kilograms is going to represent more than the muscle is capable of moving. So we apply some sort of electrical stimulus to the muscle begins to contract, tension within the muscle builds, but we can see that the muscle doesn't shorten and the load does not move. So our graphs down here show us tension within the muscle building, but the length of the muscle does not change. The muscle does not shorten. And since the muscle doesn't shorten, we do not move the load. This is an isometric contraction. Remember back in chapter 3 when we talked about iso? Well, iso meant equal. So isometric is equal length. Before and after tension within the muscle builds, the muscle stays equal length. Now let's look at some more structural layout, some anatomy of the muscle again, and this will really tie in to our muscle lab. Some of the terms are really going to overlap with lab. What we're talking about here are motor units. Here we've got our muscle and we've cut it so that we can see inside, and we've pulled out individual muscle fibers, individual muscle cells. And this red line here, this represents a motor neuron, a neuron that is innervating some of those muscle cells. And then this purple one is another motor neuron, and it is innervating some different muscle cells. Now, if we look closely at the red and the purple, the motor unit 1 and the motor unit 2, we can see if we look at the muscle cells that they are attached to, the red and the purple are not attached to any of the same muscle cells. There's 3 different muscle cells that the red is attached to. There are 2 different muscle cells that the purple is attached to, but none of these cells have a red and a purple attached to them. So when we talk about motor units, a motor unit is one motor neuron and all of the muscle cells that they innervate. So here, motor unit 1 is this purple motor neuron and these 2 muscle cells. Motor unit 2 is this red motor neuron and the 3 muscle cells that they attach to. So what happens when we are trying to contract a muscle? If an electrical stimulus, if an action potential is sent through this purple neuron, motor unit 1, then the 2 muscle cells that they attach to are going to contract, but none of the other muscle cells will contract, only the 2 that motor neuron 1 is attached to. If instead we send an action potential through the red neuron, through motor unit 2, then the 3 muscle cells that it innervates are going to contract, but none of the other muscle cells will. It's also possible to send action potentials through both motor unit 1 and motor unit 2, in which case all 5 of those muscle cells would contract. So we can actually activate different motor units within one muscle. And when we do that, we get different strengths of contractions. We could, in this case, contract 2 muscle cells. We could contract 3 muscle cells, which would give us a stronger contraction. Or we can contract 5 muscle cells, which would give us an even stronger contraction. Now, there are many motor units per muscle. So we can actually really control how strong of a contraction we get by activating certain motor units. We'll come back to that in just a little bit. Here we see a graph showing something called a muscle twitch. Now, before we actually talk about this graph, I want to talk about what a twitch is versus what a twitch is not. A twitch is not what you typically call a twitch. This is not when your eyelid is having some sort of spasm or when you get a cold chill and your body shakes. That is not the twitch that we're talking about. A muscle twitch is actually a laboratory phenomenon that does not naturally happen in the body. This is showing what does a muscle cell do in response to one single action potential, one single electrical stimulus. It's showing us the different stages of a muscle contraction. And there are 3 stages that we need to know about. First is this dark blue shaded area here called the latent period. The latent period is the time from arrival of the action potential up to release of calcium and binding of calcium to the troponin, ending when the cross bridge forms. The next period is the period of contraction. This begins at formation of the cross bridge and goes all the way until detachment of calcium and breaking of the cross bridge. Finally, we get the period of relaxation, and this is when the sarcomere goes back to its resting length. So latent period, arrival of the action potential, release of calcium, binding of calcium, and formation of the cross bridge. Period of contraction, we're going through the cross bridge cycle, tension is building within the muscle. And then period of relaxation, calcium is being pumped back into the SR, the cross bridge has detached, and the sarcomere is going back to its resting length. That is one single twitch. That's the result of one single electrical stimulus or one action potential. This graph is still of a single twitch, the result of contraction from a single stimulus, but it's showing us about how quickly that happens, and it's showing us the tension that builds within that muscle cell. So here's when the stimulus arrives, and then we go through all the way back to relaxation, and that takes just shy of 100 milliseconds. So this is extremely quick, but again, this does not normally happen in the body. This is a laboratory phenomenon just to show us the details of a single muscle contraction resulting from a single stimulus. In the body, we do not get single stimuli. We get multiple stimuli, one after another after another. But when those multiple stimuli come, we actually have different results based on how quickly those stimuli come. Here we see the red arrows representing when a stimulus is sent to a muscle cell. The blue line represents tension within the muscle cell. So the first stimulus comes, we go through a twitch. But before we come all the way back down to relaxation, another stimulus comes. So we go through a little bit more contraction. Tension builds. The actual tension is the same for both, but since we're starting from not relaxed, since we're starting from a period of partial contraction, when the second stimulus comes, we actually get to a stronger state of tension. And then the same thing again, we begin to relax and then another stimulus comes, so we contract even harder, building more tension. Relax a little bit, and the next bit of the next stimulus comes, contract even more, building stronger tension. And that was it. The tension stops because there are no more stimuli. We go back to rest. Here, the stimuli have come fast enough that we did not ever go all the way back to rest, but there was a little bit of relaxation just before each new stimulus was applied. The result is something called unfused tetanus or incomplete tetanus. Here, tetanus does not mean the disease. Tetanus is strong muscle contraction. That is not the same thing as tetanus the disease. We'll talk later about what tetanus the disease is. But unfused or incomplete tetanus results when a stimulus is applied before the muscle completely relaxes. So more tension builds. But if the stimuli come really fast, if the stimuli are like we see here, each stimulus arising before the muscle has any chance to relax at all, instead of that kind of wavy line like we saw before, we get something that looks like this graph. The muscle contracts very strongly and stays contracted until the stimuli stop, and then it goes back to its resting length. This is called fused tetanus or complete tetanus. Complete tetanus results when the stimuli arrive so quickly, when they're so frequent that there is no point of relaxation at all between one stimulus and the next. So you get a very strong contraction and a very smooth contraction. Now we've used the word tetanus twice, so I want to compare that to tetanus the disease. Tetanus the disease can actually cause tetanus the way that we're using the word here, strong muscle contractions. But tetanus the disease comes from a bacteria typically found in the soil or in rusty metal. So this is a botulism bacteria. So it's botulinum tetani or something like that. I'm not a micro guy, but it's related to botulism that we've heard about with food poisoning and stuff. So the botulism that causes tetanus actually gets into your motor neurons and causes contraction of muscles. Usually we think about this in the jaws, because another name for tetanus, the disease, is lockjaw. It affects lots of muscles, but we tend to hear about it with the jaws, the masseter. And tetanus causes you to bite down so hard and you can't relax your jaw, and your teeth meet, but you keep biting down, and you bite down so hard that eventually your teeth shatter, but you keep biting down, and that is tetanus, the disorder, lockjaw. But tetanus, as we're using it here, this is not the disorder, the disease tetanus. This is tetanus muscle contraction. So let's relate the strength of a stimulus to the strength of a contraction and to motor units. These three graphs go together, and they can be a little tricky until we understand exactly what it is that they're trying to show us. But up at the top, these red arrows represent the strength of stimulus. So this is how quickly our action potentials arriving at a muscle. Remember, that's how we actually determine strength when we're talking action potentials, is frequency. Now, when the strength is a very small stimulus, which is not enough to activate any of our motor units, we don't get any muscle cells contracting, so no tension builds. If the stimulus is a little bit stronger, well, still not enough to activate any motor units, not enough to activate any muscle cells to contract, and still no tension builds. Stronger still, finally we have reached enough to actually activate some motor units, a couple of muscle cells contract, and we get a little bit of tension. As the stimulus gets stronger and stronger, we activate more and more motor units, so tension builds and builds and builds, until eventually the stimulus is strong enough to activate all of the motor units. So all of the muscle cells within that muscle contract, and we get the strongest contraction possible from that muscle. We could still apply stronger stimuli to that muscle, which is what we see here with 8, 9, and 10. These are stronger stimuli. But at number 7, we already activated all of the muscle cells. So even though 8, 9, and 10 are sending stronger stimuli, there's no way to activate more than all of the motor units. You can't contract more than all of the cells within a muscle, so the strength of contraction, the tension within the muscle cells, do not increase. It can't get any higher because we've already contracted all of the muscle cells. So that's how stimulus strength relates to motor units and tension or strength of contraction. In this graph, we're actually looking at the size of the muscle cells, the size of the muscle fibers, and how they relate to tension or strength of contraction. Here we see a very small muscle cell that has a certain tension, but a larger muscle cell is able to contract with more tension, provide more contractile force. A very large muscle cell is able to provide a lot of contractile force. So the larger the muscle cell, the greater the strength of contraction. But the other thing that this graph is showing us is that when we're doing motor unit recruitment, when we are adding more and more and more motor units to get a stronger contraction, the first motor units that are recruited have very small muscle fibers. Then we add new motor units that have larger muscle fibers. And finally, at the end, we add the last motor units that have the largest muscle fibers. So not only are we adding more motor units, each successive motor unit has larger fibers, larger cells. And that is how we add more and more strength, by adding more motor units and larger muscle cells. Next, I want to talk about muscle tone or tonus. Muscle tone and tonus, those words are interchangeable. Tonus is the state of low-level contraction of all of the muscles in your body. You are never fully relaxed. Even when you are at your most relaxed, even when you are asleep, unless you have some sort of nerve damage, then your muscles are always slightly contracted. And that goes back to motor units. You have many motor units per muscle. And at any one moment, some of those motor units are contracting while others relax. And then some of those will contract while the others relax. So there's constantly a little bit of stimulation going to your muscles, resulting in a low-level state of contraction all the time. And that doesn't stop until death or some sort of nerve damage like paralysis. We measure this in lab during the muscle lab. It shows you that even though the muscles in your arm are completely relaxed, we can actually detect a low level of contraction in your hands, in your arm. So to sum everything up that we've talked about, what things actually increase contractile force? What things give you stronger muscle contractions? First, more muscle fibers. The more muscle fibers, the more muscle cells that contract, the greater the strength of contraction. Large muscle fibers. The larger the size of the muscle fiber, the greater the strength of contraction. High frequency of stimulation or wave summation and tetanus. We saw the greater the frequency of stimulation, the greater the strength of contraction. And finally, when a muscle is slightly stretched, it will contract harder. So muscles that are stretched just over 100% of their resting length, those muscles will contract harder than unstretched muscles. And that's what we see here in this graph. We see sarcomeres that are either not stretched, stretched to full resting length, or overstretched. And we can see down here the blue line, that shows us strength of contraction. A muscle that is unstretched, a sarcomere that is not stretched, does not give us a very strong contraction. That's because, remember, the actin, those are being pushed into the H zone towards the M line. Well, if the muscle is not stretched, if the sarcomeres aren't stretched, there's not really a lot of room to shorten these sarcomeres. But if we stretch a muscle first, here it shows it 100% stretched, which in reality, if we stretch it more than that, if we stretch it about 120%, so that the actin is beginning maybe here and here, well, there you can actually get a greater strength of contraction. That's because now there's much more room for that actin to be pushed towards the M line. So a muscle that is stretched 100% or just over 100% gives you a greater strength of contraction. But over here on the right, we see once we get the muscle stretched too far, that contractile strength goes down significantly. And that's because, as we see in this picture, the myosin cannot really grip the actin. Most of the heads are unable to grip any actin. So we don't get a very strong contraction from that. If this slide doesn't make perfect sense, please be sure to either message and ask about this or to ask about it at our next in-person meeting, because this actually comes up several times in Bio 139, because this actually applies to all muscle. It applies to skeletal muscle, which is what we're discussing, but it also applies to cardiac muscle, and it applies to smooth muscle. When muscle is stretched, it will contract harder. So make sure that makes sense and that it's something that you kind of file away in your memory where you can access it easy again next semester. Very, very briefly here at the end, I want to talk about some smooth muscle and some cardiac muscle information just to compare and contrast to what we've seen in this lecture with skeletal muscle. Smooth muscle is involuntary. All of the muscle we've talked about so far with skeletal, that is all voluntary. We think about moving those muscles. But smooth muscle, we don't think about. That's something that our body does without any conscious input. When we look at smooth muscle under a microscope, it is non-striated. It does not have any of that stripy appearance when we look at it. And that's because it doesn't use sarcomeres. Since it doesn't use sarcomeres, we don't get those bands like the A band and the I band or the Z disc or the H zone. Those are not present in smooth muscle. The shape, we call the shape spindle-shaped or barrel-shaped, as we saw back in chapter 4. Smooth muscle cells kind of look like an oval that somebody pinched the end of, and there's a single nucleus in that muscle cell. We get, instead of a motor neuron ending at a motor end plate with axon terminals, we're actually going to have something really different here. Smooth muscle is controlled by your autonomic nervous system, and autonomic nerves have a really different structure at the end. Instead of branching out and having axon terminals, we get these swollen areas near the end, and these are called varicosities. Varicosities really act very similar to axon terminals. They still have synaptic vesicles that have a neurotransmitter inside of them, but they're not just at the end. They kind of spread out, and they're all along the ending length of the axons. And near these varicosities, along the surface of the smooth muscle, there are small pits called caveoli. Caveoli are just depressions in the smooth muscle. And in those depressions, there are special channels that allow calcium to go through. Normally they're closed, but when the neurotransmitters are released, those channels open and calcium actually comes into the cell from the outside. Yeah, maybe a little bit of calcium might be released from the sarcoplasmic reticulum, but we don't really talk about that calcium. In smooth muscle, the vast majority of calcium actually comes in from outside in response to those action potentials from the autonomic nervous system. Once inside, the calcium binds to something called calmodulin. Calmodulin kind of acts the same way as troponin. It allows myosin and actin to interact with each other. This myosin and actin, even though they're not going to be in the same structural layout because they don't have any sarcomeres, they're still going to behave the same way. The actin and the myosin will attach to each other, and the myosin will move the actin. But instead of contracting end to end the way that skeletal muscle does, here we've actually got this kind of mesh net-like structure that surrounds the smooth muscle cells. And when this actin and myosin interact with each other, we actually get the muscle cell contracting in on itself from all directions, end to end, front to back, top to bottom. So we go from this relaxed smooth muscle cell to this contracted smooth muscle cell. Now, cardiac muscle kind of has a little bit of the same properties as skeletal muscle and a few of the same properties as smooth muscle. And again, we're not going to talk about this very much, but it will come up next semester in Bio 139 where you actually study cardiac muscle in depth. Cardiac muscle is involuntary, just like smooth muscle. We don't think about it contracting. It contracts without our conscious input. It is striated the same way that skeletal muscle is striated, and that's because it does use sarcomeres. It contracts end to end the same way that a skeletal muscle cell does. Unlike a skeletal muscle cell, though, it's branched. It's kind of Y-shaped, and the muscle cells are very short, and they join end to end at intercalated discs, as we talked about back in chapter 4. Calcium, still maybe a little bit of it will come from this very underdeveloped sarcoplasmic reticulum, but most of the calcium comes into the cell from the outside. So that's it for muscle activity. There are some disorders on your outline. Those are self-study. You should be able to find them in your textbook, or you might be able to just Google them and read about them online. They are fair game for an exam. We're just not devoting lecture time to them. But if you have any questions about them, feel free to let me know. Our next lecture is the central nervous system. When you look at your outline for that, you will see that it is very brief. It does not cover much of the chapter at all. I'm just going to talk about some of the more well-known structures in the brain and tell you what they do.